Okay, once again, at a synapse, right? You've got the first neuron, that gets depolarized to threshold here at the axon hillock. It'll initiate, an action potential. You'll open all those voltage-gated sodium and potassium channels, and the depolarization, repolarization will propagate all the way down to the axon terminal, right? And this is the presynaptic membrane, which is, studded with voltage-gated calcium channels and also filled with membrane-bound vesicles that are themselves studded with, transmembrane vesicular transporters, that utilize ATP, that convert that chemical energy of breaking that, ATP bond into, kinetic forcing against the concentration gradient force of neurotransmitter inside each of those, vesicles. Remember, there's a whole set of docking complex proteins that hold these individual vesicles, towards the very front of the membrane here, in close opposition to the actual, presynaptic terminal membrane. And the arrival of that, depolarization, that action potential, swings open the voltage-gated calcium channels, allows for calcium to, come in, driven by the concentration gradient and electrical forces. It interacts with, a chemical which interacts with the proteins that make up the docking complex, and the vesicle fuses, and then the neurotransmitter leaves because there's a hole now, from the tightly packed, vesicle filled with neurotransmitter, out into the cleft, which has, far less neurotransmitter. And that process by which the neurotransmitter sort of spews out of these vesicles into the cleft is known as exo, which means exiting, right? Cytosis, which means the cell. So exiting the cell. And it turns out it's kind of interesting. The vesicle, the docking complex is, complicated. There's like 28 or so proteins, right? It brings the membrane of the vesicle, close to the axon terminal membrane and causes fusion. So there's a hole now, so that the neurotransmitter can spew out. They can have exocytosis, but those proteins keep moving, and then the vesicle is pulled back into the body of the cell and sort of released, so it can go back and get filled with more neurotransmitter and get work its way up to the front again, and then spew out exocytosis again. So the process that opposes exocytosis, the release of neurotransmitter from those vesicles, the pulling back of the vesicle, is called endo, which means body cytosis. So it's pulling it back into the body, basically of the cell. And that's important if you think, because if you kept adding the vesicle membrane to the actual presynaptic terminal membrane, it would get bigger, and it would bulge out and cause all sorts of problems. So these vesicles are typically reused. Endocytosis follows exocytosis. Now, postsynaptically and presynaptically, we have receptor proteins. In this class, we're going to focus more on the postsynaptic receptors, but the types of receptors I'm going to be describing here actually are found both presynaptically and postsynaptically. So while there's often a postsynaptic density, like a pretty thick protein thickening of the postsynaptic let's say dendritic membrane that's going to be receiving the chemical message, because there's so many receptors that are arrayed postsynaptically at a synapse, at a chemical synapse, there are still receptors that are localized presynaptically as well. So the presynaptic sending neuron, the one that's releasing the neurotransmitter, right, is also listening, to the chemical release of neurotransmitter basically at, a synapse. In addition, remember the astrocyte, whose membrane wraps around the synapse and maintains a little bit of that, point-to-point chemical signaling integrity, also expresses, receptors in its own, phospholipid bilayer, that of the astrocyte. And remember, other portions of the astrocyte reach out and wrap around capillaries. So the astrocytes are listening to the amount of chemical release of neurotransmitter, the amount of activity, at a chemical synapse, and often responding, if there's a lot of stimulation of their receptors, with the release of chemicals that provoke the expansion of the capillaries in that specific region of the brain to help support that enhanced activity. So postsynaptically, we have a lot of these receptors, but there are also many of these similar kinds of receptors are located presynaptically. and also on the astrocytic membranes. But there's going to be a number of important subtypes of receptors. The strength of the chemical synapse is the fact that a single chemical that's released can actually have more than one impact, more than one effect on both the presynaptic, the postsynaptic, the astrocytic cell membranes, because it can act simultaneously on all of these different subtypes of receptors. So, for example, serotonin is a very specific neurotransmitter that, when released, has up to 15 distinct subtypes of serotonin receptor that it can bind to and provoke changes in the structure of, which can then have very specific sort of functional effects on both pre and postsynaptic cells. So I want you to look at some of these images, and we're going to start talking about the different kinds of receptors. But I want you to think about at least two types that we're going to be, two classes of receptor that we're really going to focus on in this intro class. One type of receptor, one class of receptor does this. Here's the receptor, right? And here's a neurotransmitter that's been released, let's say, presynaptically. This is a postsynaptic receptor. The neurotransmitter comes over, it binds. There's going to be a change in shape, right? But the change in shape is going to result in the opening, the ungating of a channel that will let ions pass through, okay? So we're going to see that this particular class of receptor that binds a neurotransmitter and then ungates a channel within its structure is called an ionotropic receptor. It has other names too that we'll talk about, but ionotropic receptor versus another kind of receptor. So this is another class, right? Here's the receptor, let's say, in the membrane, in the postsynaptic cell, and here is another chemical attached on the inside. So my head is here on the intracellular space, right? And when the neurotransmitter binds to this receptor, it also will change shape. But the consequence is not the opening of a channel, but it's going to be the release of this protein that was formerly coupled to or attached to, this particular receptor. We call these metabotropic receptors. They're also called other names. They're called G-protein coupled receptor. We'll talk about these names in a moment. But the protein that is now released because of the binding of the neurotransmitter is now free to go off and interact with other chemicals inside, the postsynaptic cell in this case, right? And can thus influence the metabolism of that cell. So these are two very important classes of receptor. They're found actually both presynaptic, postsynaptic, and even on astrocytic membranes. But we're going to talk about them in terms of the interpretation of the chemical message at a synapse by the postsynaptic membrane, by the next neuron in the network. And these include the ionotropic receptors that open channels in response to the binding of neurotransmitter, and the metabotropic ones, the ones that shift their shape in response to the binding of neurotransmitter, but release a protein that can then go off and influence